Okay, thank you very much, David, for this brilliant talk. You know, it was like honey for me. You know, just listening. It's so great. Thank you very much, David. So now we move to the next speaker. So the next speaker is Vivian Williams. Vivian is CEO of SME company based in Dublin. The company is called Cellix. So Vivian, the floor is yours. Do you want to use this microphone? Okay. So you have two hands, two tools. Okay. Can we have the next? Okay.
Perfect. Okay. Okay. So first of all, I'm just going to give you a bit of an overview about Cellix, and then I'm going to give you an example of one product that we've been trying to bring to market. So Cellix was started in 2006. We're based in Dublin in Ireland, where we have manufacturing facilities, a research lab, and so on. We're backed by both Irish and international VC. One of the VCs is based in Paris, called OTC, and so far we've raised just under €3 million, which by comparison to some of our competitors or other people in the field is not too bad. We have developed microfluidic pumps, biochips, primarily for cell-based assays. Okay, but the market is small, and so we noticed that we had to re-engineer some of the things that we were working on and start to work on integration of detection techniques on chip and start to target new applications. Our existing product portfolio is sold directly, but primarily through a network of distributors, and you can see some of the distributors here on the map. So these are the products that we sell. You can see the chips on the left, in the middle the pumps, and through the expertise that we gained in developing these products, as I said, we started to integrate detection techniques on chip, and we've developed now this INISH device, which I'm going to tell you about today. So we developed it as a hematology analyzer, and the real reason we started in that space is because we got funded by the European Commission to do so, which is always a good starting point. But when we started here, we really needed to talk to some of the people in the field, because ultimately we knew when we started that we weren't going to be selling this device. We were going to have to partner with somebody who could sell it for us, because it was not in a field that we were familiar with. We sell to research labs in both industry and academia. So we started to talk to people in the field, some of the large multinational companies, and what they said to us was that, yes, there is a market for a smaller device in this field. You can see some of the devices here from Abbott, Horiba, Sysmex. These are the kind of standard mainstay devices in this field. There's a new device that's just been launched on the market from Samsung. And one particular company said to us, Vivian, if you can build us the exact same instrument that Samsung has, we'll take it. So they wanted to compete with them. And this is a sizable company. So the reduced footprint was going to be key. They want to target emerging markets, and particularly they're looking at things like ER, ambulance, war zone situations, so near patient testing. For this, they said the device can be less sensitive than the standard-based instrument, but typically you would have CVs of less than 3%. Okay, so that's what we were looking at trying to develop. So in terms of market drivers and needs, then what we needed was a simple, user-friendly, compact, benchtop flow cytometer which could target POC markets, which is basically what everybody else wants. So you know, the standard simplification of your currently complex flow cytometer. So as I said, we're looking at emerging markets. There's a couple of barriers to the market, and one of them was obviously the market maturity. Some of the companies that I mentioned, like Sysmex, Abbott, they're really well established. So we've gone to speak with companies like Abbott, Sysmex, and so on, that are equally well established in order to find a partner for access to the market. One of the other restraints is IVD certification, ISO 13485. Something which is difficult for small companies like us. We have CE marked our products before, but we haven't gone down this route. We haven't needed to. So in terms of commercialization strategy, it's, as I said, heavily dominated by large multinational players. So we were looking to partner with one of these players so that we could bring the device to market. And our strategy was to sub-license the device. It was then called the DIMID Plus device, and that was because it was under that was the name of the European grant that we had, the FP7 grant. And to either sub-license the device for the hematology field, or to look at supply of the device on an OEM basis. So in terms of our key requirements, we needed a portable device, small footprint, had to be low cost, sales price had to be ideally less than €10,000. Some of the devices I just showed you earlier are typically in the region of 16 to 25,000. We had to have disposable chips or reagents because there had to be some sort of razor blade model for the company to continue sales with. And it had to be capable of RBC counts, so red blood cell counts, WBC counts, which is white blood cell counts, with 3-part differential. But it didn't necessarily have to be as sensitive as your lab-based instruments. So CVs didn't have to be as low as 3%, but near, as close as possible. So the Hematology Analyzer, here it is, it's built, it's done. So it's fast, reliable, label-free, which is one of the most important things. You don't have to use any dyes. And it can be used for detection, characterization, and viability studies. It's portable, it's low cost, we can run it off a 12-volt car battery. And we actually made it as sensitive as the Sysmex XP300, so we do have CVs less than 3%. And we also benchmarked it against BD's Curie device. Just a little bit of background, it's based on impedance spectroscopy. This is a blown up picture of the chip. And what happens is you scan frequencies simultaneously, and you can actually look at not only the size of the cell, but you can look at the cell membrane, so you can easily do viability studies. But you can actually even go further than that. You can actually look at cytoplasm, vacuole content. So it's an extraordinarily sensitive technique, which can be used for a wide range of applications outside of just hematology analysis. We can also detect 5,000 events per second. That's really important in terms of dealing with your sample. And we can do lots of other things. I won't go into detail about that, but the important thing is that in terms of throughput, it's competitive with what's already on the market. So this is the white blood cell counting. For those of you not familiar with this field, you've got lymphocytes, monocytes, and granulocytes. Those are your 3 different white blood cell populations, okay? So we had to go and benchmark the device and show these potential partners that it did actually work. So we benchmarked it against the Curie, as I said. Here you can see we got CVs of less than 3%, 2.71% in fact, so pretty close. But obviously we're selling this for 3 times less than the Curie, so that's a significant advantage. We also did the viability studies with it, and again, got comparable results. Okay? I'm not going to go through all this data in detail. All I'm going to show you is just some of the graphs to show you the benchmarking study against the Sysmex XP300. But again, we took healthy volunteers, we took samples and looked at RBC counts, WBC counts, lymphocyte, monocyte, granulocyte percentages, counts, and so on. And in each of these, we were getting CVs of less than 3%. In some cases, as you can see on the RBC count, around 1%. So very, very close to a well-validated product on the market. This is the WBC count, the lymphocyte percentage, CV of 2%. Here, slightly off, but we had one outlier. And CVs of less than 3%, and so on. Okay? So I suppose, we went back then to our partners, and we said, okay, here's exactly what you said you wanted. We had been in touch with them all the way, and they were really, really favorable to the results. They said, yes, we really like what you've developed. They had real faith in the data. We're very impressed with the CVs, particularly as they didn't expect that we could get them for a portable device. And they didn't seem too concerned that we hadn't got certification in place. And that was something I was concerned about because I knew that was going to take a lot more money than we had at the time. But what we did notice was the goalposts had moved. In the time that we had started the project to the time that we had delivered the results, which was March of this year, the industry has moved. So what they're looking at now, what they want now, is a 5-part differential. So instead of just looking at lymphocytes, monocytes, and granulocytes, you see the granulocytes there in the first box. They now want to actually see a 5-part differential. So they want to see eosinophils, basophils, and neutrophils as well. So this is a little bit more tricky and a lot more work. So they had kind of moved the goalposts on us, which was a bit disappointing. That being said, they are still interested, and we're still moving ahead with demonstrations on site with this potential partner. So you could see this on the market as a hematology analyzer, but it won't be under our name. So what next? In the meantime, we've spent a lot of time working on this device. So there was no way I was going to sit there and say, right, well, you know, they're a bit slow. It's going to take probably another year or whatever by the time we get certification, get it up and running with one of these partners. What else can we do with it? So we started looking at beer. And why wouldn't you? And so in beer, one of the things that's really important is yeast. And in beer, we wanted to look at the number of yeast cells per milliliter. We had one particular customer who is one of the top breweries in the world, and what they want to do is actually have a portable device to count the number of yeast cells in a sample taken from the tap. So they want to go into a bar, they want to take a sample, put it through our device, and tell you that that is below a certain number in terms of yeast numbers per milliliter. What that means is that their line won't get contaminated, the keg won't get contaminated, and it saves them money. So in one area, for example, in one country, they would expect to do probably 40,000 tests per annum, just testing bars ad hoc once per quarter. So it's quite a nice application, but it's also really, really simple for us. For us, it's just a simple viability study. We had to do absolutely no development. We just put the sample through the device and it worked immediately. So they were delighted. One other area that they were interested in, but not as important for them, because it doesn't affect their business as much, but one area that they were also interested in was bacteria. This is important for me because it actually applies to a number of other application areas that we're looking at. So bacteria, if you're not familiar with microfluidics, you can't use a pulsatile device for looking at bacteria. They don't like it. So you need really, really good microfluidics, which if you read all the documents that the European Commission send out, microfluidics and nanofluidics is one of the key enabling technologies that they all want you to look at. So that's where we operate, and that's where this device has one of its really, really good strengths. And so that allows us to look at bacteria, where a lot of other devices, and particularly some of the devices on the market that are using kind of cheap peristaltic pumps, they can't do this. So we can look at bacteria, we can do live and dead studies, but we can also do an awful lot more. So this is one area that we're looking at in more detail. There's also a couple of other areas that we're looking at. In fact, last week we took delivery of a very nice sample of Pinot Noir from the French Institute of Wineries, and they are particularly interested in looking at subpopulations of different cells, of different yeast cells. So you see, you can actually start to extrapolate there, look at different application areas where we can sell the device. I think the hematology analysis will come eventually, but it's slower. And so in the meantime, we've invested a lot of time, effort, and money into developing this device, and you can be sure that we're going to make sure that we sell the device for something. Biofuels, pharma and cosmetics, in conjunction with things like droplet generation, which is another area that we work on, is something that we're also doing, and we're in contact with a number of companies in these fields as well. So our partner, our next steps in terms of commercialization is to continue to look for partners for market access for the supply of the devices, either on a sub-licensing or OEM basis. We can demonstrate the device on request to any interested party, and we're going to continue to actually validate the device per application area to show that it actually really works there and it can be used in the field. So if anybody does want to talk to me about that afterwards, by all means, you can ask me during the session or come talk to me afterwards, and there's my contact details. Okay, thank you very much.
Very good. Well, thank you very much, Vivian, for this very nice